You'll notice a few changes already. You'll find that I do not use PowerPoint for my lectures, and there's a reason for that. I'm doing it as a favor for you for two reasons. First of all, there's a malady in academia that I call death by PowerPoint. There is no more efficient way of putting a room full of people to sleep than droning on through PowerPoint slides. The other point is that your careers will not be available for you on PowerPoint slides. We live in an age where you are flooded with information. It is not hard to find information per se. You are drowning in it. And the principal challenge that you and I will face for every day of our careers is figuring out how to filter all that information. What is important, what is not important. And having everything available for you on PowerPoint slides denies you the opportunity of trying to figure out what is important. Each day I will have all of my lecture notes on the boards before the class starts. The order will always be 1, 2, 3, 4, 5, 6, 7, 8, and sometimes, like today, there'll be a bit more information on a 9th, and God forbid, even a 10th board, but be aware of that. You are welcome, of course, to take digital images of the boards, but that kind of defeats the purpose because I didn't really want you to spend the time on PowerPoint slides, so making your own, you can do it, but recognize that that's perhaps giving you a liability. Instead, I would very strongly urge you to use the webcasts as a backup tool. The webcasts are produced at a substantial expense. We're fortunate in having a grant from the Howard Hughes Medical Institute to fund part of that effort for this class. So I strongly recommend the use of webcasts. Now, I'm going to have the pleasure of being able to follow Dr. Dowda. Yes, sir, you have a question. So the problem with the webcast is that it is blurry, and that's why the audio is pretty good, because any word that you can't read that is on the board is a word that I will be saying as clearly as I can, though with my upstate New York accent, that may be a challenge from time to time, too. But please stop me like that anytime that I say something that's not clear. So thanks. Okay. As I was saying, I had the pleasure of following Dr. Dowden, who's given you the foundation, which will make the subsequent discussions, I think, more fun. Because you have a vocabulary now to work with, you have concepts, and you have some methods to work with, and the next 5 weeks will be much more experimental. So we'll be dealing with what is the experimental evidence for how we know something. It is not so important what we know. What is important is that we know it. So science is not a collection of opinions and ideas and deconstruction of this or that. It's a foundational set of truths that have survived experimental validation. And what I really want you to be able to understand is when a scientist say that we know something or we're pretty sure, how you know the level of confidence in that. So we'll deal a lot with experimental evidence along the way. I'll try to de-emphasize the amount of memorization that you need to do, but there is some. Biology, as you've already discovered, is in fact a foreign language. And the best way of becoming conversant in a foreign language is to converse in it. So I very strongly urge you to routinely get together in groups of your colleagues and talk about what we learned in class. An exceptionally good way of learning is to try to write exam questions. Because there's a limited amount of material in any lecture that I will be able to ask an exam question on. And so as you discuss the material with your friends speaking the language of biology, it will become second nature to you. And the more exam questions you try to write, the more of those you'll probably hit that will be the same exam questions that I will write. I will also be periodically posting, as I already have, thought questions that are relevant to the topic of the discussion. So there's about 12 or 13 questions that are relevant to today's discussion that's on B-Space. Take a look at those. If you can solve those problems or think through those problems, you'll be very well positioned for the kind of exam questions that I'll ask, and you'll get copies of past exams. And trust me, I am a person of extremely limited creativity. The exam questions that you will get from me this year will not be the same exam questions that I've ever given before, but they couldn't be all that different because of this limited creativity. So among the things that you must memorize, if you haven't done it already, and I hope you have, is the alphabet. You have to know the order of letters from A to Z, and the reason you have to know that is that the index in your text and every other text you will ever use is arranged in alphabetical order. So there will be things that I absolutely will be expecting you to get from your text. Only some of those will be listed on the reading pages for each lecture, and I'll try to point out those that are hit or miss here and there, but I'm going to expect you, for example, in today's discussion, to go to the index and look up transposable elements, and you will find background information that's pertinent to our discussion today that's sort of scattered over a bunch of different pages in different editions, okay? So be familiar with the index in your book. So I'll start our discussion of the second third of BIO1A with really one of my favorite topics, and that's the issue of microbiology. You have heard, no doubt, the saying that the meek shall inherit the earth. I think that's nonsense. It is the microorganisms that are going to inherit the earth. The cockroaches probably have a chance too. Microorganisms are the most diverse form of life on the planet, and microbiology is the study of all single-celled organisms and their viruses. Let me move my cell phone. Now, microorganisms are of two types. There are the prokaryotes, so named after the Greek for no nucleus, and a subset of the eukaryotes. The eukaryotic microorganisms include protozoa. They include fungi, one of my favorites. We'll talk a lot about a certain fungus later in the class. And the prokaryotes broadly break down into two different kingdoms. There's the eubacteria, the so-called true bacteria, and an entire kingdom of life that was not discovered until Carl Woese recognized it in the 1980s. These are the remarkable and extraordinarily versatile prokaryotes called the archaebacteria. And one of the subtexts I hope that we'll always be thinking in the back of your mind is that when I tell you about what we know, I want you always to be thinking about what do we not know. And it still astonishes me that when Leeuwenhoek in the 1600s first recognized some of these kingdoms, that an entire kingdom of life on Earth eluded our best efforts to detect it until 1980. We wonder what else we're missing. Surely a lot. Now, the number of microorganisms is really astonishing, and if you were to take the dirt under your fingernail and scrape out the amount of dirt under one fingernail and use a method that we will talk about on Friday to take a census of the number of different species in that amount of dirt, you would find that there is about 10 to the 5th species in that amount of dirt, okay? Assuming that the dirt is from soil, for example. All right? That's astonishing. There are only 40,000 vertebrates in all of zoology, different species, 10 to the 5th different species under your fingernail. Now, we sometimes use the royal we, but in fact, we're never really using the royal we when we refer to ourselves. Each of us is made up of about 10 to the 14th human cells, give or take, and the average person, even after a shower, has 10 to the 15th bacterial cells on and in you. So they are a normal part of the ecosystem, which is us. So the idea that bacteria are bad comes from a very small set of bacteria and the problems that they'll cause. We'll end our discussion with them today, but bacteria are absolutely essential for normal health of most of us. Without bacteria in your gut, you are not as healthy as you are with bacteria in your gut. Now, one of the other things that's interesting about bacteria is their range and versatility over evolution. This is an unbranched evolutionary tree of life on Earth. And what we recognize is that the first forms of life from the divergence of eubacteria and archaebacteria occurred about 10 to the 9th years ago, a billion years ago. So at the time that the Vishnu schist in the American West was being laid down, that was when the first single-celled microorganisms started to evolve, and it was at that time that the archaebacteria diverged from the eubacteria, and then out on the branch that led to the eukaryotes that we're more familiar with, the charismatic macroflora and fauna and the protozoa, that's way out here, but way deep, 10 to the 9th years ago, is when the split between archae and eubacteria occurred. The archaebacteria you have probably seen direct evidence of. For example, anyone who has flown into the Bay Area on a plane and has circled San Jose has probably seen these purple-colored salt marshes down in the South Bay. The color of those salt marshes is purple due to the photosynthetic membrane of the halobacterium, a kind of archaebacterium that can grow in extremely high concentrations of salt. Those of you that have driven down I-5 past the feedlots in Coalinga, which a colleague of mine calls Cowschwitz, have seen Avogadro's number of cows out there turning grain into hamburgers. Well, in the guts of these ruminants, there are archaebacteria called methanogens. Guess what methanogens do? They make methane, a greenhouse gas. And agricultural production of methane is estimated to be responsible for about 16% of greenhouse gases, and methane is one of the worst greenhouse gases. On a good day, one of those cows, while awaiting oblivion, is producing 100 to 500 gallons of methane in the form of cow burps and cow farts. Okay? So think about that. As you drive down I-5, there is a major source of global warming gases. Now, because we understand the nature of that, it is possible for us to take our understanding of microbiology and start to think about what we could do to cow feed to reduce the production of methane. If we gave them a better electron acceptor in their anaerobic metabolism than CO2, they would produce something else instead of methane. So we can start to think about ways of using microbiology to really address really significant problems. Another kind of archae, some of my favorites, are the ones that grow in boiling water or in the steaming vents on the side of volcanoes. These are thermophiles. And each and every one of those proteins in those thermophiles has to be able to maintain its stability under conditions which would turn normal proteins into a gelatinous mass like a fried egg white. So there's some really incredibly interesting things that are going on in these microorganisms. Now, what thing that frustrates me about discussions broadly about microbiology is that almost everything I'll tell you is not absolutely true. It's a generalization because there's such diversity that there's an exception to everything. But generally, bacteria are small. Okay? They are on the order of a micron. You can just about see them with light microscopy. And your text has some spectacular electron micrographs of different kinds of specialized stuff inside different species of microorganisms. They can be modal. Okay, they can move in some species up to 50 times their length in a second. Now, there is not an Olympic sprinter that can move 50 times the length in a second, although in the downhill events you can certainly achieve that speed, but bacteria do it all the time. David Zussman in the MCB department and the late Dan Koshland, for whom Koshland Hall is named, were pioneers of studies of different kinds of motility in bacteria. They are generally structurally less complex. Less often do they have internal membranes, like we will discuss at length in eukaryotic cells, but they do have some. Typically, they have one chromosome, which is a circular DNA molecule, a few million base pairs long, and many of them will have small pieces of DNA, circular autonomous mini chromosomes that go by the name of plasmids, and you've already encountered this discussion of plasmids in the lab class, and we'll manipulate them later. Now, the most important thing to keep in your mind about bacteria is what Stan Falco of Stanford says of bacteria, which is the goal of every bacterium is to become bacteria. They are the champions of converting energy in any form, either reduced carbon or chemical energy, into more bacteria. And we'll talk about the kinetics and the processes by which they do that next. Let's pause and see if there's any questions. I want to encourage you to interrupt me with questions at any point. How are we doing so far? Yes, what's your name? Diana, what's the question? Singular versus plural. It comes from the Latin. Is the neutral singular, and is the neutral plural. That's all I remember from 3 years of high school Latin. Other questions? Okay. Let's move on. So here is a really lousy diagram of a bacteria that doesn't represent any bacterium in reality, but it's a way of thinking about it. So I'll work my way from the inside out. So in the inside of bacteria, there is a bacterial chromosome and all of the proteins that are associated with it, and that's called the nucleoid, or the nucleoid body. That's here in the center. There is the cytoplasm full of capacity for making macromolecules, and you've learned about some of those in an overview from Professor Dowden's lectures, ribosomes, RNA, nucleic acids of various kinds. We'll talk a lot about those in subsequent lectures. There is an inner membrane which limits the cytoplasm, and outside of that, there is a cell wall, typically in many bacteria made up of a peptidoglycan layer. It's part peptide, part sugars, okay? And then in certain bacteria, the so-called gram-minus bacteria, there is an additional outer membrane. The bacteria that have the outer membrane, as I said, are called the gram-minus bacteria. Those that have the membrane, that have the outer membrane are the gram-minus. Those that lack the outer membrane are the gram-positive, right? And the reason that is interesting is that there is a commonly used stain in medical microbiology that distinguishes about half of the bacteria on the planet as gram-positives and half are gram-negatives, and that corresponds to this physical difference. Okay. Now, a typical bacteria, as I said, is about 1 micron in length. And one of the important things to keep in mind is that bacteria divide by binary fission, not by mitosis, and they do it really fast. So if we start with one bacterium, for E. coli, for example, a typical laboratory pet, in 20 minutes we have 2, in 40 minutes we have 4, in 60 minutes we have 8, and here is something else that I would like you to memorize. I would like you to memorize convenient powers of 2, 2 to the n, where n is a digit that is 10, 20, 30, or 40. Why? Because it helps you develop a rapid intuition about what's going on in an experiment and gives you a sense of scale. So memorize that 2 to the 10th is 1,000. Okay? You could also calculate it, but sometimes memorizing things is just more fun. Now, if you remember your rules of exponents, you will therefore immediately know that 2 to the 20th is about 10 to the 6th, and 2 to the 30th is about 10 to the 9th. So why is that useful? If we start with one bacteria, they divide by binary fission in a kind of growth that we call exponential, where every cell gives rise to 2, so the exponent is 2. After 10 cell divisions, we'll have 1,000 cells. After 20 cell divisions, we have 1,000,000 cells. And after 30 cell divisions, we have 1,000,000,000 cells. And at 20 minutes per cell division, you can calculate how long it would take to grow an arbitrarily large number of cells. It's very useful to develop that as a sense of scale. Now, any collection of cells that comes from a single cell is called a clone. And so a colony of bacteria that comes from a single cell, like the Vibrio colonies and the other colonies that you've already grown in the lab, is a clone of cells. In the case of E. coli or Vibrio, a single colony that you could see with your eye, especially the Vibrio that glow in the dark, it's about 10 to the 9th cells. So it's a very large number of cells, and we'll do some experiments with that number in just a second. Now let's imagine that we were to take a tube of broth, food for bacteria, and to inoculate that tube of broth with about 10 to the 3rd bacteria per mil in that tube, and simply monitored the increase in the number of bacteria over time. This is a growth curve, a so-called growth curve. We're plotting the number of cells over time. And if we measure the number of cells by optical density or any other spectrophotometric method, we would see that after a while, if we took cells that were stationary phase, that had stopped growing, and put them in this fresh broth, that there would be a period of time where cell number did not increase. This would be the time during which the cells are sensing their environment, they're importing the nutrients, they're getting the production lines of macromolecules in order. We'd then see the optical density start to increase, and there would be a period of exponential growth in which each cell is dividing at the same time with the same growth rate as every other cell. And then as they eat up all of the nutrients, converting mass into bacteria, they would enter stationary phase again. And typically, anytime you do an experiment in a lab, you use cells that are in the exponential part of the growth curve in order to be sure that every single cell is in the same physiology so that the measurements that you make of that culture represent what's going on in all cells in that culture. However, we're beginning to recognize that the bacteria that we are interested in on the planet are often not in exponential growth phase. They are in biofilms on catheters, for example. They are lining the bottoms of ships, decreasing their efficiency of going through the water. And so there's a whole new branch of microbiology, this sort of non-exponential growth phase microbiology, which we're discovering some fascinating colonial properties of bacteria. And in fact, the Vibrio that you've experienced in the lab, when they reach a certain culture density, have a property that changes. They kind of accumulate and populate specialized organs in squid so they can produce light. So bacteria have some really interesting colonial and population properties. Now, as I said, bacteria have, on average, typically one large chromosome. The size of the chromosome depends upon the bacteria. E. coli is probably about average. And it has 4.3 times 10 to the 6th base pairs in its entire chromosome. Another number that you might want to try to remember is that the typical protein is about 50 kilodaltons, and it takes for a typical gene a little over 1,000 base pairs to encode a typical protein. And so if you know those numbers, you can say, there's about 4,000 or so proteins that are probably encoded in a bacterial cell. Some bacteria get by with quite a bit less than that, but this is typical. About 4,000 proteins, a little over 4 million base pairs in the genome. Now, in every division, though, the entire genome has to be replicated, and DNA replication of a bacterial chromosome starts at a particular place called the origin of replication, and it proceeds bidirectionally all the way around the chromosome, replicating every single base with remarkable fidelity. There are errors which occur during replication. They lead to changes in the genome called mutations, and their frequency is about 10 to the minus 9th per base pair per generation. Okay? So that's pretty amazing. How many things can you do with that level of precision? Right? For me, the answer is none. Okay? But now that lets us think about what it really means to be a clone. Let's pause for a second and consider the following. Suppose we start with a single cell and grow that cell to a population of 10 to the 9th cells. How many, and that population conveniently fits in a nice small-sized tube, how many mutations have we accumulated during the growth of that culture? Think about that for a second. See what you can do with that problem. So we end with 10 to the 9th cells. Okay? How many mutations did we accumulate in that culture, assuming this is the error rate? Anybody come up with an idea? Let's see. Douglas Forbes, are you here today? Douglas Forbes? Jay Kumar, how about you? Are you here today? Ooh. Do I have this year's class list? How about Lawrence Lee? Lawrence, you here today? Lawrence, work with me on this. How many mutations do we get in that culture? How would you approach the problem, Lawrence? Okay, so let's, Lawrence, work with me on this. So if we end up with 10 to the 9th cells, and we have 4 times 10 to the 6 base pairs per cell, how many base pairs do we end up with in that population? You had calculus yet? Okay, right. So we 10 to the 9th, right? times 4 times 10 to the 6 per cell. So we end up with 4 times 10 to the 15th base pairs. And our error rate was how many mutations per base per cell division? What was that? You remember? 10 to the minus 9th. Okay, so we'll take this number of base pairs times 10 to the negative 9th. And that's going to equal what? Mutations. Really? That's the number of mutations that happened in the last cell division, because it was the last cell division that gave us the 10 to the 9th cells. So 10 to the 9th cells takes 30 cell divisions, so we have to add this number of mutations to the number of mutations that occurred in every cell division before that. Is that right, Lawrence? So how many cells were in the previous cell division? Half of 10 to the 9th, right? So what would that be? Five times 10 to the 8th, right? So at that same rate, we would end up with half that number of mutations having arisen in the previous cell division. So it'd be 2 times 10 to the 6th from the previous cell division, and it'd be 1 times 10 to the 6th from the previous cell division. So by the time we get done adding this up, the numbers that we're adding get smaller and smaller, of course, but it gives you some sense that a clone of cells that came from a single cell with this fantastic precision of DNA replication is, in effect, a population of cells with a genetically hedged bet against what the future will be. Because as the environment changes, some genotypes, some collection of mutations will be more fit, others will be less fit, and the more fit ones will be able to outproduce the others. So just because it's a clone, do not get the idea that they are all identical. And the development of genetically different individuals in a bacterial population will become a big deal as we get to the last few boards. How are we doing? Any questions on that? Yes, sir. What's your name? Ken? Ken. Well, why were we including the number in the previous cell division? Because the mutation, once it occurs, stays except for when the error rate turns it back to what it was to begin with. So we can assume that the rate of reversion of a mutation is low, as is the rate of formation, but once it exists, it persists. So essentially, we get to add all the mutations that occurred during the history of the development of that culture. Okay? So why is the biosphere not all bacteria? Right? And the answer is that bacteria have their problems too. And one of their problems is the limiting amount of food. Okay? And the other problem is something called bacteriophage, sometimes abbreviated phage, and sometimes abbreviated by the Greek letter phi. I think of bacteriophage as being software in search of hardware. They were discovered by two not particularly nice people, Frederick Twort and Felix Durrell. It was a spectacular discovery, but no Nobel Prize was given for this because they weren't very nice. But there was a Pulitzer Prize given to Sinclair Lewis for his novel from 1926 called Aerosmith, which if you haven't read it, I highly recommend it. It's terrific. It's a not very disguised discussion of Durrell's life. Describes the discovery of bacteriophage. Now, what was Durrell doing? Well, Durrell, because he was not a nice guy, was sent by his boss from the public health system in France to the Yucatan of Mexico, which in 1915 was not exactly Cancun, okay? So he was the public health official there, and during the time when he was there, the Yucatan was infested by a locust plague, okay? They were eating the guys out of house and home, literally. But he discovered, he'd heard in parts of the Yucatan that there was something killing the locusts. This is an incredibly bad drawing of a locust. And what he found oozing from that particular end of the dying locust were bacteria. Bacteria that he called coccobacilli, and he deduced that it was the coccobacilli that were killing the locusts. So he started growing the coccobacilli into one of the world's first examples of biological warfare. He was going to spray these coccobacilli all over the place and stop the locusts. So if you plate a bunch of coccobacilli on a Petri plate, put about 10 to the 6th on the plate, you won't even be able to see them. But if you incubate that plate overnight, you discover that the plate becomes covered with a whole lawn of these bacteria, most of the time. But sometimes he discovered that there were zones on that plate in which every single cell had become lysed. He called those zones plaques. So what he was able to show is that there was something in this plaque that was actually killing the bacteria. The way he did that was to take these plaques and dilute them again and again. And he reasoned that if there was some toxic agent, that the toxic agent, when it got diluted enough, would no longer be toxic. But what he found was that when he diluted a piece of agar from this part of the plate and then plated that diluent onto fresh lawns of bacteria, that he got plaques that were just as big. So they were capable of amplifying itself. Okay? And he was also able to show that whatever it was that was so-called undilutable was filterable. It was smaller than a bacterium. It was getting through the finest filters that bacteria could not go through. So he discovered then the very first examples of viruses that grow on bacteria. And if there are 10 to the 5th different bacterial species under your fingernail, I can assure you there's more phage than that. Okay? So generically, what a phage looks like is a tiny little thing that is a protein coat inside of which is packed a phage chromosome. A phage chromosome, depending upon the species of phage, can be as small as 5 genes. That's about the smallest that I know of. And there are recently some that have been identified by Graham Hatfill in Pittsburgh that have up to 1,000 genes. Okay? Now, the protein coat that surrounds the phage chromosome has at its base a motor, a DNA pump that was characterized by our colleague here at Berkeley, Carlos Bustamante, that burns ATP to pump DNA inside the head of the virus. It is extremely pressurized here. The phage chromosome is packaged under 60 megapascals of pressure. Anybody have an idea about what 60 megapascals is like? Anybody here ever opened a champagne bottle? Pop, right? A lot of pressure there. That is 6 megapascals. So the pressure that is on the bacterial chromosome packaged inside a phage head is about 10 times the pressure in a champagne bottle. Okay? So that's really quite remarkable. They burn a lot of ATP to do that. Now, why do they do that? That puts the chromosome in there like a wound spring, so that when the phage then encounters a bacterial cell, as soon as it makes contact with the surface and there's a little rearrangement to make an opening, zing, the bacterial cell is infected by the phage chromosome. Okay. Now, there's 2 lifestyles of phage, bacterial viruses. And this also applies to human viruses, 2 kinds. There are the so-called lytic viruses, like phage T4, which when it infects a cell, it immediately takes over the replicative capacity of the cell to make more phage parts. So here's the infected cell with the bacterial chromosome. Over time, the phage parts, the heads, the tails, and the chromosomes get made independently, and then later on, they spontaneously assemble into new phage particles, and then genes in the phage make essentially detergents that lyse the cells. So one micro investment, destroyed environment, maximum yield for the phage. If that sounds like the behavior of investment bankers these days, I agree. Okay. There are, however, more sophisticated phage, as typified by one studied widely called lambda. And lambda has the capacity of sensing its environment. So, for example, if lambda infects its chromosome into a cell that's a happy cell, full of energy, it does the same thing as T4. It goes lytic and produces lots and lots of lambda. However, if it manages to find itself infected in some E. coli that's strung out on a poor carbon source, not much energy, what it does is to integrate its chromosome into the bacterial chromosome. Because being part of a bacteria is a better survival strategy in nature than being part of a phage particle. As that bacteria replicates and divides, then the bacterial replication of the chromosome replicates lambda's chromosome as well. And when one of these cells then gets back into a good state of health, the phage switches to a lytic pattern and makes more of itself and lyses the cell. Okay? And we now know a great deal about the molecular mechanisms of that switch from a lytic state, from a lysogenic stage to a lytic stage. We really understand that in great detail. There's some vocabulary words here. The lysogen is the word that's given to the bacterial cell that harbors a quiescent phage. That quiescent phage is described as the prophage. Okay. Questions so far? Yeah, what's your name? Ming.
So when the phage is incorporated into the part of the chromosome of the bacteria, does it also undergo mutations with the bacteria when it replicates?
It's a great question. Is the prophage susceptible to the same mutational processes that happen on the bacterial chromosome? Yep. The phage will endure mutations at the same 10 to the minus 9th per base pair per generation. And you can do a calculation for a phage that's 5 genes long, 1,000 bases. What fraction of the prophages would be mutant after a certain amount of time? Good question. Yes? This is for bacterial viruses that we're talking about. Phage is the word used for bacterial viruses. Animal viruses and plant viruses are called viruses. Okay. So let's talk about sex, which in bacteria does not involve anything like love, and I'll show you why, but it's really all about the exchange of DNA. And we'll see that bacteria make a lot of effort to try to have safe sex, to exchange DNA only with their own species. We'll discover some unsafe kinds and the significance of that for us. So with respect to sex in bacteria, there's really 3 kinds. There's a kind called transformation, a kind called conjugation, and a kind called transduction. And I'm going to leave it to you in your text to read about transduction. I won't cover it in lecture per se. But we'll start with transformation. Why? Because it's an incredibly useful tool in the lab, and it also gives me the chance to highlight the accomplishments of a former Berkeley undergraduate, Hamilton Smith, who in 1981 got the Nobel Prize in Medicine and Physiology for his understanding of how bacterial transformation worked. This is the first of the Berkeley undergraduates that I will tell you about who got a Nobel Prize. There'll be many more, and we will be expecting that of you as well, so don't get lazy. So Ham Smith made his career studying Haemophilus, the kind of bacteria that gave you ear infections for which you drank that disgusting pink amoxicillin that your parents stored in the refrigerator. If this brings back flashbacks and nightmares, I'm sorry about that. Now, what Haemophilus wants to do is to be able to check with other Haemophili to see whether any of them, when they died, had a better genetic idea than that Haemophilus has. That is, did it have any variant in its genes that would be better off being transmitted to another Haemophilus? So here's the Haemophilus with its chromosome, and here's some Haemophilus DNA from an old dead Haemophilus, and here is some foreign DNA that might be a virus DNA. So what Haemophilus has is actually a receptor on its surface that recognizes an 11 base pair sequence that is common in the homophilous chromosome, but not common other places. And when it finds that, it brings that DNA into the cell. It does not bring foreign DNA into the cell. And then by a process that we'll talk about later on in the course, recombination, its genes can be swapped for the incoming genes, and if those genes are better, that homophilous outproduces the other homophili. Okay? So this is a mechanism of recognizing self from non-self outside the cell, minimizing the risk of taking up unsafe DNA. Most other species of bacteria rely upon a different mechanism for recognizing self versus non-self. This mechanism is commonly referred to as the restriction modification mechanism, using restriction enzymes and modification enzymes, and I'll outline it broadly with this example here. So here is a sequence that you might recognize as the same spelled this way as it's spelled that way, so it's a complementary sequence. Such sequences, short sequences in a species-specific way within a species, are recognized by enzymes called methylases, which decorate particular positions, either an adenine or a cytosine, with a methyl group on that sequence and no other sequence. Such bacteria also produce an enzyme, a restriction enzyme, that will cut that sequence if and only if it is not methylated. So a bacterium like E. coli, if it is infected by a bacteriophage that did not come from E. coli, that phage will not have these methyl marks, and its restriction enzyme will destroy the DNA of that incoming phage. So the whole industry of biotechnology has been driven by the discovery and development of enzymes like restriction enzymes that allow molecular cloning of genes. It all came from self-protection mechanisms in bacteria. Okay? And there are more than 100 different kinds of restriction enzymes, each of them specific to a particular species. I'll move on now to conjugation, another form of bacterial sex. This was discovered. Whoops, where is conjugation? This was discovered by Joshua Lederberg when he was a student at Stanford. Lederberg was not the first Stanford student to discover sex, nor was he the last, but he was the only one to receive a Nobel Prize for his efforts. At the time that Lederberg discovered the sex of bacteria, it was thought that bacteria did not have sex. He discovered two kinds of bacteria, though. Those that had the fertility plasmid, those were called F+, and those that did not. So he was working with E. coli, and here is an E. coli cell with its bacterial chromosome, and here's another E. coli cell with its bacterial chromosome. This one has the F plasmid, this one does not. The F plasmid is called a conjugative plasmid. It lives to move itself from one cell to another. It encodes a protein, several proteins including a pilus, and the pilus actually assembles into a channel connecting two different bacterial cells. When this channel is established, DNA replication, which we'll talk about a lot on Friday, makes a copy of the F plasmid and transfers that copy into the recipient cell. And this connection is very tenuous, so mild agitation separates these two cells in which the donor has donated its F plasmid to the recipient. Okay? Now, you may wonder why there are any F- bacteria in the world, and that is because cells with the plasmid grow a little bit slower than cells without the plasmid, so there's a selection for those that manage to lose it. And also, life's not fair. For the so-called F+ or male bacteria, there are phage that attack specifically them by recognizing the pillin proteins that make up the pilus. So there's kind of an arms race going on in the microbial world. Now, the other really important thing about conjugation is that although Lederberg studied it in E. coli, conjugation is completely non-consensual sex. It is so non-consensual that a bacterium will mate its DNA of the conjugative plasmid variety, not just into many different kinds of bacteria, but into plant cells and animal cells. And we will see that it is this ability of bacterial conjugation to move a DNA from a bacterium into a plant cell that is the foundation of biotechnology with agricultural applications. Essentially, all plant genetic engineering takes advantage of this wild and woolly sex. Now, this year, for the first time, I'm taking a risk and introducing to you another crazy kind of DNA that may cause conceptual challenges for us later, but I'm going to take that risk and talk about transposable elements. And what a transposable element is a piece of DNA, not very long, that contains really just 2 genes. One gene makes an enzyme called a transposase, which allows this piece of DNA to jump. It jumps from one place to another. And the other gene, or genes, are genes that control resistance to drugs. So imagine the following. Let's imagine we now have an Enterobacter faecalis with its F plasmid that has a drug resistance transposon in its F plasmid. If it mates to Staphylococcus aureus, that mating can result in the hopping of the transposon off of the F plasmid into the S aureus chromosome. If the Enterococca were resistant to a drug that the Staphylococcus is sensitive to, this non-consensual sex can immediately give rise to drug-resistant Staph aureus. Okay? That's bad. Why? This is penicillin. This is the first line of defense that has kept most of us alive over the years. It was discovered in 1920 by Fleming, synthesized by Fleury, and penicillin inhibits a cell wall biosynthetic protein that binds to a peptidyltransferase that makes that peptidoglycan layer. If you are resistant to penicillin, then you no longer are capable of being cured by this kind of an antibiotic for an infection. Now, there have been many lives of synthetic organic chemists spent on changing this R group to some other R group to make derivatives of penicillin, which allow us to combat cells that are resistant. So let's take a look at this timeline. In 1942, 1/2 of the world's supply of penicillin was spent to cure one person of a bacterial infection. This is in the middle of World War II, and by 1945, thanks to the government's investment into pharmaceutical companies, that number went to 646 billion doses from one in the course of 3 years. So penicillin got widely spread in the world, providing enormous killing potential of bacteria and the development of resistance. And in deference to the time, and because it's a very important point for those of you that will think about medicine as a career, I'm going to stop our discussion today and pick this up at the beginning of next time. Okay? So next time will be DNA replication, and I'll finish this evolution of drug resistance in bacteria for you. My office hours are listed in the syllabus. I don't actually remember where they are, but you can find them there. And be sure to look in B space for the problem sets to think about.